All right. I believe we finished the conversation on lipids. I think that's what we talked about on Monday. Yeah. So far, we covered two biological molecules, two biological macromolecules, biological polymers. We covered carbohydrates, and we generally covered lipids, triglycerides, phospholipids, and sterols and hopanoids. Now the next two big classes are the ones that we're going to talk about a lot. Those are proteins, and then we're going to chat about nucleic acids. So let's talk about what proteins consist of.
We're going to work on your doors out there. We're going to work on your door right here. The door handle's really loose. You all right with that? All right.
So proteins consist of 21 different amino acids. Now, this statement sometimes can be a little confusing. You have to understand that protein can have 100, 150, 200, I don't know, 1,000 individual amino acids. But there are 21 kind of different amino acids. Is that clear? So you may have learned before that there are 20. No, there are 21. And we'll tell you when time comes to translation, I'll show you that there's 21. So those are different amino acids. There are a few conceptual things that I want you to notice to understand about those building blocks of proteins. If you would look at these 6 different amino acids, and as I mentioned, you do not need to memorize any chemical formulas. What I want you to understand, look at these parts. You see that these parts are all the same. You have an amino group, you have a carboxyl group, and you have central carbon. And you will find it in any amino acid in the human protein, whether it's lysine or glutamine or aspartame. What differentiates those amino acids are the side chains. In this image that I'm erasing from now, side chains are highlighted in blue. Can you see those? Methyl group here, thiomethyl group here, in cysteine, and so on and so forth. Okay. Now, why it's so important? You can imagine that if side chain has different structure, it will also have different type of chemical interaction with other side chains. Do you understand what I'm trying to say? Think of amino acids as 21 different pieces of Lego. And depends on how you arrange them, the final structure that you get is going to be different. Does that make sense? So far, does that make sense? You sure? We're good. Okay, because we're getting there. So, if you take one amino acid and another amino acid, 2 amino acids, in the reaction of dehydration again, they form peptide bond. So amino acids are connected to each other in the peptide or in the protein by the peptide bond. Are we clear about it? Now, you can imagine that you can just keep adding amino acids, forming eventually a chain. Here in the left bottom corner, you see a chain of amino acids, what we call a polypeptide. And we can know the exact sequence of amino acids. This is called primary structure of a protein. The exact sequence, just the sequence, is the primary structure. Now, each amino acid isn't just a dot. Each amino acid is a pretty large molecule with side chains sticking out, and depends on what the side chains are, they will determine how this chain that you see in the left bottom corner, how it will fold. Does that make sense to you? Do you understand what I'm trying to say? Okay, think about this. Imagine that you have a rope. Okay, can you, I don't know, make some sort of a tight, can you wrap it tightly around some kind of a, I don't know, cylinder? Now take barbed wire. It's going to be much harder because it is shaped differently. It has those things sticking out. So this is what I'm trying to tell you, that depends on which side chains amino acids in the sequence have, that will determine how they're going to fold. And the first folding here is called secondary protein structure. Secondary protein structure has two main different elements. Proteins usually form either alpha helix or beta sheets. Okay, and you can look at the protein structure and say, aha, we can see here alpha helix, we can see here beta sheets, and each structure has certain functions. Let's put it this way. And then these helices, the blue one, and the sheets, the red one, can fold even further. Okay, and they will form what is called a tertiary structure, kind of a globule. Okay, now this tertiary structure of a protein, for many proteins, that's it. This here can be your protein molecule. I'm trying to think of an example. Well, a lot of transcriptional factors and translational factors, they are just one single polypeptide. Okay, insulin is a polypeptide. Okay, hormone like glucagon is a polypeptide. Single molecule. Bless you. Sometimes, though, proteins consist not of one polypeptide, but of several. Okay, something like Voltron. Yeah, very few cultural references here to Voltron. Never mind. It's a giant robot that consists of 5 smaller robots. There was a cartoon that was very popular, Japanese cartoon, very popular when I was a kid. Really? I gotta rewatch it. So it's ridiculous, stupid. But I was 6. I have an excuse. So, for instance, a protein called RNA polymerase, protein that synthesizes RNA in bacteria, it consists of 5 different subunits, 5 polypeptides, 5 globules like that. Does that make sense? So 5 subunits. Each subunit has its own tertiary structure, but when they come together, they form a functional protein. You take one subunit, it is useless. Only by putting them all together, it's going to work. Okay? Now, so try to use your imagination and think about this. You synthesize somehow a polypeptide, and you let it go, and it starts to fold by itself. And it eventually assumes this conformation in tertiary structure. Okay. Thank you very much. Sorry. It's all right. Now, can we move that backwards? Does that make sense? Can we make proteins to go from tertiary structure to secondary, from secondary to primary? Can we make the structures go backwards? The answer is yes. This process is called denaturation. I used to use as an example of denaturation the cooking of an egg. Egg is mainly protein, but I have a better example, really. I like it more. So, hair procedure, like, say, hair straightening, okay, or doing a perm. Both of this is the protein denaturation. You have a protein called keratin in your hair. And different people have different conformations of this protein. Some people have straight hair, some people have curly hair. Now, if you say have straight hair, you go to a hairdresser and they apply a bunch of chemicals and a temperature to denature keratin and give it a shape that they want to. Your hair becomes curly. But later, that treatment goes away because keratin in your hair will slowly assume its normal shape. Does that make sense? Same way it goes with the hair straightening. You can straighten the hair, but eventually they will become curly again because keratin in your hair will assume its normal shape. So protein gets denatured and then it renatures. Unless you completely break down the primary structure, it can come back. When you cook an egg, you break down primary structure as well. It's not going to come back. Okay. Speaking of function, and that is really important, I want you to understand that life, like all manifestations of life, for bacteria, I don't know, whether or not they have flagellum, whether or not they're resistant to antibiotics, whether or not they can use, I don't know, certain sugars as the nutrients. All these manifestations of life, things that we call together a phenotype, are because of proteins. Does that make sense? Say, if bacteria can have flagellum, flagellum consists of proteins. If bacteria are resistant to antibiotics, they have some kind of an enzyme that makes them resistant, some kind of protein. If there is a function, this function is 99% cases is performed by a protein. That's the function of a protein. If we think, okay, this bacteria can, I don't know, live in a salty environment. Why? Because it produces proteins that, I don't know, pump the salt in, pump the salt out, whatever. But it's a protein in the end. Am I clear? Now, the second really big class of molecules that I want to mention are nucleic acids. So we're going to, later in the course, in the second unit, we're going to pay really close attention to two of them. DNA and RNA. So DNA is double-stranded helical molecules. All DNAs in a cell are double-stranded helix, like this one in the picture. There are 4 types of nucleotides, individual building blocks that form DNA. You all know it, A, G, T, and C. They do form complementary pairs. A pairs with T and G pairs with C. And this is what's holding DNA molecules together. That pairing, those nucleotides in the opposite strands, they interact with each other, forming hydrogen bonds, and hold the strands together. Now, DNA, what is the function? DNA carries the genetic information. Okay? And I'm going to give you my analogy that some people told me works pretty good in explaining the functional relationships between DNA, RNA, and protein. So, to make analogy, to give it more sense, let's mention what the function of RNA is. Main function of RNA is serve as a messenger. Now, to analogy. Imagine that in your family you have a book with recipes, meal recipes. And this book is your family heritage. There are recipes that were handwritten by your, I don't know, grandmother. So it's really a treasure, your family treasure. This book is enormous. It contains all kinds of recipes, from absolutely outdated that you will never cook, to modern, to rare. You may have, I don't know, 15 different recipes for apple pie. You have thousands of them. Understand? That's your DNA. Oh, crap, that's embarrassing. I don't remember the correct spelling. Is that it? Now imagine that you expect somebody to come over. And people that come over, you have a vegetarian, person with a gluten intolerance, and a couple of people that can eat everything. So you think, what am I going to cook? Probably going to cook something that is gluten-free. Okay? You're going to have some vegetarian options, and you're going to have some animal protein options. Chicken, poultry, whatever, fish. And since it's a 4 people, we're probably not going to make like 20-course meal. So we're talking about 5 dishes maybe. So you need 5 recipes, right? It's a freaking 20,000 recipes book. And it's old and it's your family heritage. You're not going to bring it to the kitchen, are you? You can spoil it, you can burn it, all types of bad things can happen to it. Okay? Now, how can you deliver those recipes to the kitchen? What do you think? Take them out. Well, you can spoil them and then it's gone, huh? Okay, yeah, you can write them on a piece of paper. I'm lazy, so I will just snap a picture with my phone. Either way is fine. Basically, the book stays in your, I don't know, living room, your bedroom, wherever you store it, okay? You keep it as a treasure. That's your DNA in the nucleus or in the nucleoid. And then you take those pictures, specifically of the recipes that you need, and you get them to the kitchen. In this case, pics of recipes, that's RNA. Okay? And this process right here is called transcription. DNA to RNA is transcription. You basically make kind of a copy. Does that make sense? The whole idea. But you can't feed your guests with the pieces of paper with recipes written down on them. You really need to cook. So use these recipes to make meals. And that's protein. Because in the end, what you put on the table is food, not the recipe. Make sense? In the end, what does all the cellular work are the proteins. Not the RNA, not the DNA, it's proteins. But in order to get to them, you have to start from the protein recipe, from a gene. So I deliberately used 20,000 genes, 20,000 recipes. Human genome has about 20 to 30,000 genes. But every given moment, only few of them are expressed. Only few of those recipes are actually used to cook, quote-unquote, cook meal, make proteins. Does that make sense? Same is true for any living organism. Now, keeping this analogy in mind, let's think about properties of DNA. First of all, DNA is double-stranded. Each strand carries the entire genetic information about the organism. Why do you need 2 strands? You have one strand that carries the entire genetic information, and another. What is the function of the second? You have one recipe book, and then in the basement you have second one. a copy. That's a copy. That's a backup plan. Okay? If page is missing, you have a copy in the basement. Does that make sense? So, that's one thing. Second thing, this book, is that like, are you going to throw it away? Are you going to destroy it? No. DNA is super stable and very well protected. Now, those pictures on your phone, or those pieces of paper with a recipe, what you going to do when you're done cooking? Yeah, you're going to get rid of those, right? You don't need them. Same goes for RNA. RNA messengers, they thrown out. You don't need them, really. Does that make sense? Okay. Now that brings us to a slide that I'm really proud about. Two cornerstone concepts that we're going to use in this course over and over again. First concept is the concept of gene expression, what we just talked about. Now it's easy for you to understand what we're doing here. So this, your recipe book, that recipe book, in the recipe book of bacteria, all the genes, we call it a genome, right? Genetic information about the entire cell. Okay? Now, you know that expression, genotype, defines phenotype? I mean, it's good, but I want to make a bit of a correction here. E. coli, you know, heard about that. E. coli, recent outbreaks with the romaine cellar. E. coli has about 5,000 genes. Not all 5,000 genes are expressed at a given moment. Meaning, E. coli does not cook all 5,000 meals that it has recipes for. Only genes that are essential to that particular moment are expressed. And gene expression at the given moment defines phenotype. Let me give you a really easy for me example that is really easy. You have E. coli. Normally it uses glucose as the source of carbohydrates. Makes sense. You follow me, right? You take E. coli and you put it in the medium where there is no glucose but a lot of lactose. E. coli starts to express proteins that can break down lactose and use it as an energy source. So, environment, changing environment, lactose instead of glucose, changes the gene expression in E. coli. Does that make sense to you? And we can say, okay, we had this E. coli that were able to grow glucose. Now we have this E. coli that can grow lactose. Phenotype of the cell has changed. Think about this. In the summer, if you spend a lot of time out in the sun, you get tanned. The reason? Protein called melanin is produced at much higher rates. Environment changes your phenotype. Your physical appearance is changed. Does that make sense? The only big difference between humans and bacteria, or viruses for that matter, protozoa, humans change phenotype much slower, or at much smaller level, because we reproduce really slow. Does that make sense to you? So kind of, sort of take home message from here. Phenotype is determined by gene expression at the given moment, and phenotype are observable or detectable traits. So what can be observable or detectable traits for bacteria? Tell me. Speak up. Size, good. What else? They not really colored, too small for it. Shape. Huh? It got to this size. I just mentioned. Okay, flagella. Great. Whether they can move or they can't move. Okay. How many flagella they have. I mentioned lactose. They nutritional, properties. That's observable. You put another bacteria on lactose and it dies. It can't use lactose. That's a different phenotype. Does that make sense? Now, we're going to transition to another important, really important phenotypical trait in bacteria in a second. If you change the environment, organisms will change the gene expression, the phenotype, in order to adapt to this environment. For humans, it's hard. If tomorrow there will be a law that all people who are taller than 3 feet will be executed, majority of humanity will go extinct. It's really hard to adapt overnight and become 2 feet 10 inches, right? Bacteria have a certain advantage. They reproduce extremely fast. And as they reproduce, they accumulate mutations. Some mutations can be bad. They may not be compatible with the survival. Other mutations can be what I use this word, good. They will increase the fitness. They will give these particular cells an advantage over others. And here comes one of the most important things in a modern medicine, antibiotic resistance. So imagine that you have a population of bacteria. Blue cells represent regular microorganisms, and this orange cell represents bacteria that are resistant. They express some kind of a protein that gives them that resistance. Now think about this. If there is no antibiotic in the environment, being resistant to antibiotic, is that an advantage? No, because there is no what is called selective pressure. I will be using this word in explanations later, so just, I want you to get familiar with that. Selective pressure. So my example with if you taller than 3 feet, you're going to get executed. That's an example of selective pressure. However, if you add antibiotic to the environment, cells that are not resistant are going to die. And cells that are resistant are going to survive, and they're going to multiply much better, to much higher rates than non-resistant cells. And eventually, you get a resistant population. Does that make sense? So, certain pattern of gene expression in orange cells gave them fitness advantage under certain selective pressure. And the phenotype of a population changed. Does that make sense? You can develop bacteria that are resistant to hand sanitizer. Not 80%, but if you dial down, last year the paper was published. Scientists did that in the lab. They dialed down on the ethanol concentration to about, I believe, 20%. And they managed to produce bacteria that can survive 20% ethanol. Which is pretty cool. Just showing that adaptability is not limitless, but close to this. So, a famous geneticist, he was an immigrant from Russian Empire, Teodosius Dobzhansky. Well, strictly speaking, he was Polish, but at that time Poland was a part of Russian Empire. He made the famous statement, a biological statement, nothing in biology makes sense except in the light of evolution. And in this course, when we're going to talk about mostly clinical events, we're going to try to keep in mind the evolutionary implications, why bacteria got to this point, or viruses got to this point, and gene expression. We talk antibiotic resistance, gene expression. We, I don't know, talk resistance to ability to grow in the presence of oxygen, gene expression. Without oxygen, gene expression. Does that make sense? So it's all about gene expression. Any questions? Okay. So we're going to talk. This is going to be really like, before it was really drowning chapter, but I threw some really cool clinical examples. So it's going to be quite exciting. And we're going to start with a discussion of bacteria and archaea. Now, I want to clarify one question. If you're going to take other biology classes later, if you decide to move on and do a 4-year biology degree, and you take junior, senior level biology classes, you may run into professors that are vehemently opposing the term prokaryotes. Now think about this. Prokaryotes contain which two domains of life? Bacteria and archaea, right? And if you remember that Woese-Fox classification, that phylogenetic tree that I showed you, bacteria and archaea, they aren't even brother and sister, they're cousins. So they're really far from each other. So many biologists embrace this idea and they say, oh, no, 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 no. You cannot put them in the same group. Well, my take is it's convenient. They structurally similar. It's just like, you know, when we talk about 4-legged animals, it's fine to put, I don't know, a horse, a cow, and a pig in one group if we need to talk about the mode of locomotion, that they all walk on 4 legs, okay? They differ, we know that. So we keep in mind that bacteria and archaea are different, but it's very convenient to talk about them as prokaryotes. Technically speaking, term prokaryotes is incorrect, but a bunch of people still use it, okay? And you're allowed to use it here. It's a prokaryote okay space. Okay. So first, the shape of the cells. This is something, sorry, this is something you need to know. I will ask you. It's really easy to memorize, but I will ask you. Coccus, round-shaped cell. Bacillus, rod-shaped cell. Vibrio, curved rod. These names are reflected in the names of some microorganisms, like Staphylococcus, round-shaped. Bacillus, like Bacillus anthracis, rod-shaped. And Vibrio cholerae, causative agent of cholera, which is curved rod. Coccobacillus, clumpy sort of in between the rod and the round, okay? And these two spiral-shaped bacteria, Spirillum and Spirochete, the difference between those. Spirillum is shorter and thicker. Spirochete is longer and thinner. That's basically it. Any questions? Example of a question. Rod-shaped bacteria are called, and you pick bacillus. Yes. So that's kind of, that's easy stuff, but I want to check if you went through this table. Now, sometimes cells, bacterial cells, can bundle together. And this is really important because that's an important phenotypical trait. For instance, when cocci form these pairs, this is called diplococci. When they form this 4-cell assemblages, that's a tetrad. Why I say that's important? When we will get to staining of bacteria, there are some species of microorganisms that can be isolated from your hands. You put it under the microscope, you look at them, there's going to be a bunch of diplococci and tetrads. And they are characteristic for that particular species. So look at them and say, that's Mycococcus, because it forms this arrangement. Okay? Streptococcus, chains. By the way, Greek word coccus means grape. So, Streptococcus, chains of grapes. Staphylococcus, clusters of bundles. And Staphylococcus is the bundle of grape. Finally, just like Streptococcus, Bacillus can form chains, and this is called Streptobacillus. Clear? Any questions about this table? That's super simple, no tricky questions, just know this stuff. You have to match the name and the description. Go ahead, your question.
I was asking because I can't see the board.
I can tell you. Which one?
The last one.
It's Streptobacillus. Streptobacillus.
Thank you.
Sure. Now, how the heck do bacteria maintain such shape? For this, we need to recall what osmosis is. It's a diffusion of water, basically. Movement of water through semi-permeable membrane. Okay? Semi-permeable membrane means that water can get through the membrane and solutes like ions, salts, cannot. Okay? So the direction of osmosis is from low concentration of solutes to high concentration of solutes. It's kind of sort of mnemonic rule here. Not so much mnemonic, but kind of, common sense rule. We call water a solvent, right? So water wants to dissolve or dilute more concentrated solutions. Water always tries to get where it's more concentrated solution to equal out the concentration. Does that make sense? Now, let's use it as let's play with some examples. So this is the first example. So you see, you have this little pouch here. This pouch has 20% solute concentration. The environment around has 60% solute concentration. So environment outside is much more concentrated than the water, than the solution inside the pouch. Where does water go in this case? Outside. From the pouch, outside. If this pouch is the cell, what's going to happen to the cell? It's going to shrink. Excellent. Okay. Now, if the cell is placed in the really, really diluted environment, so 20% inside versus 10 outside, water is going to move where? 10% outside, 20% inside, into inside. Yes, what's going to happen to the cell? It's going to get larger and may eventually explode. Does that make sense? Now, when solutions have equal concentration inside and outside, any movement? Net movement of water? No. So if you take bacterial cell, which is about 70% water, and put it in this isotonic solution, is this cell going to change its shape? No. You put it now, I'm going to not go in ABC order, I'm going to talk about hypotonic first. You put it in hypotonic solution. So concentration of solutes outside of the bacterial, say, is lower. Water is going to move where? Into the cell. What's going to happen to the cell? Get larger. But bacterial cells have a cell wall. So the cell wall will limit the swelling. That make sense? This is called turgor. Okay, cell is kind of, you know how to say it. It's really, I have no English words for it. Honestly, I have no Russian words for it. It's like slightly swollen and solid, like a good, I don't know, like a good apple, you know, like a balloon. Okay. If you will remove the cell wall, by the way, what's going to happen to the cell? It's going to explode. This is the mechanism of some antibiotics. They weaken the cell wall and cells explode. Okay. Now, you of course can tell me what happens to the cell in the hypertonic solution. It shrinks. Excellent. Look at this cell. Do you think it's happy? No, it doesn't. It's not happy. It doesn't feel well, right? Now, it sounds really fundamental, but you use this approach to put bacterial cells in a hypertonic solution, or should I say, you know, industry uses it every day to preserve food. Salty foods. When you pickle things, you usually add a lot of salt. So you put possible bacteria in this heavy hypertonic solution. Does that make sense? You know how ages ago, when people went on Royal Navy ships, British Royal Navy ships, one of the main food was very much salty pork. Okay? Because it could be stored for a long time without getting terribly rotten. It was somewhat rotten, but not terribly rotten. Okay. Now, we got this idea of movement of water, and I will ask you this. I will ask you what happens to the cell in such and such solution, just to make sure that we're all on the same page. Now, the concentration of solutes in the solution is called osmolality. So say if you take a bucket of water and pour down there a pack of salt and stir it in, you're going to have the solution with a high osmolality. If you put down a pack of sugar, still high osmolality, right? Just a high concentration of solids. Do I understand that? Now let me tell you something. There are microorganisms that can survive pretty high concentration of salt. For instance, typical Staphylococcus aureus can survive up to 7.5% sodium chloride. To give you a perspective how concentrated that is, you take a glass of water, a cup of water, a cup, measuring cup, and you add a tablespoon of salt to it. That's salty. Staphylococcus aureus can grow in this environment. It doesn't shrink. So, how can cell survive in the environment with a high osmolality? I deliberately didn't use the word isotonic hypertonic, just high osmolality. So there are a lot of solutions on the outside. A lot of solute on the outside. What should cell do to survive? Then it's dead. A lot of solute on the outside. What should it have inside? No, because then a lot of solutes on the outside. What should be inside? A lot of solutes too, exactly. So cell has to be able to increase its internal osmolality to equalize it with the environment. Does that make sense to you? Staph aureus by no means is the most brutal. We will talk about halophiles, but you know the Dead Sea in Israel? There are bacteria that live in Dead Sea. The concentration of salt, you know, it's so high that you cannot possibly drown. Your body will not ever sink. So, bacteria live in that environment. Yeah, they're pretty sturdy. Any questions? Now let's talk about cell parts that can be found in bacterial cells. There are 4 parts that can be found in any bacterial cell. Nucleoid, here. Nucleoid is cellular DNA that is bound to so-called nucleoid-associated proteins. Obviously, the function of DNA in that nucleoid is to store genetic information. Cytoplasm, that fills out bacterial cell. It's basically a storage site for everything. Okay, proteins, carbohydrates, lipids. Ribosomes. Here's the bacterial ribosome that consists of a large and a small subunit. The function of ribosome is protein synthesis. And finally, cell membrane. So this is a typical cell membrane. Cell membranes in bacteria and eukaryotes are similar. Cell membrane consists of double layer of phospholipid molecules. You can see this double layer here. In bacteria, there are going to be hopanoids. I mentioned it before. And they're going to be transmembrane and peripheral proteins. So the function of transmembrane proteins is what? What do you think? Proteins that span through the membrane.
As things in and out.
Yep, transport, exactly. Transport. Because I think we mentioned it before. Hydrophilic molecules cannot cross the membrane. It's a problem. So they need some kind of helper, facilitator. Okay, transmembrane protein. Does that make sense? Nonpolar, hydrophobic, or lipid-soluble molecules let they can go through. Does that make sense? So say ions or proteins or peptides have to have some kind of a transmembrane protein to allow them to cross the membrane. If you have an oil can get directly. Am I clear? Any questions? So this is what we just mentioned. So I want you to know 3 types of transmembrane transport. First is simple diffusion. I want to remind you that diffusion is the movement of molecules along the concentration gradient from higher to lower concentration. So diffusion doesn't require energy, since when it moves from higher concentration to lower concentration, does diffusion require energy? This is a slide. This is a ball. It goes from higher to lower concentration. Does it require energy? No. This is why we call diffusion a passive transport. Okay, so simple diffusion is the diffusion of nonpolar hydrophobic molecules. Since nonpolar hydrophobic molecules can cross the membrane directly, we call it a simple diffusion. If there is a need to transport polar hydrophilic molecules, we need to use facilitated diffusion, which requires transmembrane proteins, such as channels or carriers. in some cases, you need to transport molecules against the concentration gradient. So what you need to do is to drive that ball in the bottom right corner up the slope. In this case, the kind of transport is called active transport. And active transport allows you to move molecules against the concentration gradient. So far so good? Active transport doesn't require energy. Yes, it does. In the form of ATP. And of course, active transport always requires some kind of a transmembrane protein, what we call a pump. Any questions on the transport across the membrane? Clear? That is, we're basically doing a repetition of what you learned before. Now, I want to highlight one quite important thing. You must know 4 cell parts that can be found in any bacterial cell, or for that matter, archaeal cell. So any prokaryote has cell membrane, cytoplasm, nucleoid, and ribosomes. Am I clear? Now what about cell parts that can be found in some? We're going to start with inclusions. Kind of boring. Inclusions, bunches of chemicals. Okay, so those are all inclusion bodies. They can store hydroxybutyrate, which is a nutrient, or, like in this picture, they store iron. Bacteria live in all kinds of the environments, and they may need to store all kinds of weird chemicals, nutrients, to sulfur, to metals, often for reasons that we don't know about. Okay? What I want you to know is to know that inclusions are just accumulations of proteins in the cytoplasm. Clear? One thing that I want to remind you about, we talked about it, but I want to remind you again. In prokaryotic cell, there are no membrane-bound organelles. No organelles that are encased in the membrane. Inclusions are not surrounded by a membrane. Nucleoid is not surrounded by a membrane. This green dashed line around the nucleoid, you can see that. It just delineates it. There's no membrane there. Does that make sense? It's really important. Inside of the prokaryotic cell, there are no membranes. So far, are we clear? Now, I want to reintroduce another structure that we talked about already. Remember, we talked about Clostridia. I mentioned endospore. So, endospore is formed, anyone remembers when? More broad, when anaerobe exposed to oxygen or any unfavorable conditions. Does that make sense? For anaerobe, oxygen exposure is super unfavorable. Lack of nutrients, super unfavorable. Dryness, super unfavorable. Okay? Now let me ask you this. The top of that cabinet, or that cabinet, is it dry? What do you think? It's fairly dry. If we would go there, swab it, and put it in a agar plate, you're going to get a lot of Bacillus subtilis growing. Bacillus subtilis forms endospores. These things are covered in endospores because it's dry. Okay? And that brings us to how endospore is formed. So this is the cell. It starts to reproduce and senses conditions are far from perfect. Instead of dividing, it starts to isolate its DNA, covering it with a bunch of layers of carbohydrates and proteins, all together usually called cortex. Okay? Does that make sense? And finally, you've got a spore. You might ask, what happens to original cell? It's dead. I sometimes call an endospore an escape pod. Like, I think it was in one of the modern Star Trek movies when Enterprise exploded in the end, but they managed to escape an escape pod, which is kind of small, and they have no idea where they're going to end up. Okay, it's not a fully functional spaceship. So endospore is not a fully functional cell. It can't reproduce, it's metabolically inactive, but it can preserve DNA. Oh, yeah. There were endospores. You know what amber is, right? Amber. Like a tree sap that solidified. Scientists managed to isolate endospores from amber that are about 300 million years old. They got bad. They produced bacteria. They're fine. 300 million years old, no problem. They survived. The process of endospore formation is called sporulation. So let's write it down. You have bacteria, and you have endospore. So if you go from here to here, this is called sporulation, right? So those bacilli on top of those cabinets, when it became dry, they sporulated and formed endospores. Am I clear? Are you following me? Now you collect those endospores and put them on the agar. And endospores become vegetative, growing, reproducing bacterial cells. This backward process is called germination. Does that make sense? Like a seed. Like seed germinates in the soil or in the water. Does that make sense? So think about this. If endospore is formed in the absence of nutrients and or water, what should trigger germination? What should trigger conversion of endospore into the cell? Presence of water and or nutrients. Very good. Does that make sense? You take nutrients and water away, endospore. You add them back, vegetative cell. This is the picture of an endospore of Coxiella burnetii, gram-negative microorganism that causes disease known as Q fever. Very rare. Those are, I believe those are Clostridia, and I mean, if you will take a magnifying glass, and look at this picture, which is really, really tiny, you'll see tiny little endospores inside of the cells about to be formed. Now, why we pay so much attention to endospores? They're really hard to kill. So remember we talked about C. diff, Clostridium difficile? People shed Clostridium difficile in their stool in the form of endospores. And in the hospital, those endospores can end up in the environment, and good luck with killing them. Bleach doesn't kill them. I mean, if you take a concentrated bleach, pour it and leave it for an hour, yes, it does. But that doesn't happen. So endospores are notoriously hard to kill. Okay? There was a study, actually, of patients who suffered from C. diff. Samples were taken from the stools of the relatives who live together, members of the same household. In every household, every member of a household, not sick, they have endospores of Clostridium difficile in the stool. So it's so abundant, it's almost impossible to prevent the transmission. Make sense? So this is why we really want to know if a microorganism forms endospore or not. Now, last thing here, plasmid. So plasmid is a double-stranded circular DNA, which carries some genetic information. It is not essential, but beneficial. Does that make sense here? Think about it like you have your family recipe book. You buy specifically a book with Thai dishes. Okay, you still have your 5,000 recipes in the book, so you're fine, but you just want some extra. Okay, so this plasmid is some extra. It can carry genes that provide resistance to antibiotics, that allow bacteria to use some other nutrient source, all kinds of fancy crap. But it is not essential. You take plasmid away, cells still can reproduce. Everything's fine. That makes sense? Now, cell wall. This is one of the cornerstone structural concepts of the bacterial cell structure. There are, first of all, if you would look at the bacterial cell, okay, here, so you see this is cytoplasm, this is membrane, and this is the cell wall. So cell wall is on the outside. Am I clear? Cytoplasm, membrane, fairly thin cell wall. Okay, on the outside. Got it? So, cell wall consists of carbohydrates. You don't need to memorize these names. And peptide bridges. Look at either left or right picture, either at gram-positive or gram-negative cell wall. You see these large spheres? Those are carbohydrates. They form long chains. And these long chains are connected by fairly short peptide links. Can you see the peptide links? So essentially, you have kind of a lattice, and this cross-linked lattice provides a great deal of mechanical resilience to a cell wall. Before we move any further, do you understand what I'm stressing here? So we've got chains of carbohydrates connected by peptides. Overall, when we talk about the cell wall, we use the word peptidoglycan. So we say that bacterial cell walls consist of a polymer that we call peptidoglycan. Peptido refers to the links, glycan refers to the chains, of carbohydrates. Is that clear? Why cell wall is such a cornerstone concept? Not only it divides all bacteria into two major classes of gram-positive and gram-negative, there are antibiotics that inhibit synthesis of different components of the cell wall. Does that make sense? For instance, penicillins will prevent these links from getting formed. Now imagine that instead of a lattice, cross-linked lattice, you merely have This chains of glycans not connected by anything. This cell wall will have no mechanical resilience, right? And bacteria will explode. Vancomycin will prevent this glycans to link together, again weakening cell wall, leading to lysis of bacterial cells. So this is really like important aspect here. Now, human protein called lysozyme breaks down the links between these two units of cell wall, and lysozyme is antibacterial protein. Can be found in saliva, tears, mucosal secretions. Natural antibacterial drug. Question? If you spit on the bacterial culture, there's a good chance that the growth is going to be inhibited. That's how lysosome was discovered. It wasn't saliva. I think it was snot. Somebody sneezed, and mucus got in the bacterial culture, and there was like a huge inhibition zone. And they started to identify what that was. It was lysosome. Now, I mentioned before gram-positive and gram-negative. So here we can define what it means, and it's really important. Let's take a look at gram-positive cell walls. Gram-positive cell wall is right here, and you can see that gram-positive cell wall is a thick layer of peptidoglycan. We usually say that in gram-positive bacteria, cell envelope, not a wall, envelope, contains two layers, a wall and a membrane. Can you see that? A wall and a membrane. Okay? This blue lines spanning through the cell wall in gram-positive microorganisms are the molecules of teichoic acid. Teichoic acid can be found only in gram-positive bacteria. Only. So that is really important. It's sort of a marker for our own immune system to distinguish gram-positive bacteria from gram-negative ones. Teichoic acid can contribute to the adhesion of bacterial cells to the surfaces and generally increases the ability of bacteria to infect cells or organisms. Any questions about gram-positive? So I told you what you need to know for gram-positives. I'm going to tell you what you need to know for gram-negatives. So this is on the right, is gram-negative cell wall. I would say gram-negative cell envelope, okay? Look at the layers. Plasma membrane, and then there is a cell wall right here. You see, it is thin. It is really thin. Turns out, drugs that inhibit cell wall in gram-negatives, inhibit cell wall synthesis, they do really poorly against gram-negatives. There's not much to inhibit. Make sense? And then in gram-negative microorganisms, there is a third layer. It's called an outer membrane. So there are two membranes, the plasma membrane and the outer membrane. The membrane is attached to the cell wall through the protein called murin, these tiny little purple things. But I want to focus your attention on an extremely important structure that is unique to gram-negative microorganisms. Here in the image, you see it, it's like, you know, this red things sticking out. Can you see those? This is called endotoxin. or lipopolysaccharide. Lipo refers to lipid, polysaccharide refers to sugar molecule. So it's essentially lipid attached to a sugar molecule. You with me? Why we pay so much attention to this endotoxin or lipopolysaccharide, LPS? When you get infected with gram-negative microorganisms, at some point bacterial cells will die, and they will break apart, and they will release lipopolysaccharide in your bloodstream. And lipopolysaccharide will eventually get delivered to your hypothalamus and will reset your thermostat from 36.6 Celsius to 39 Celsius, and you will get fever. Lipopolysaccharide is responsible for general inflammatory symptoms during gram-negative infection. For instance, in people who have colitis or some intestinal damage, often gram-negative bacteria from the gut get into the bloodstream. Do you follow? And the first symptom of that infection, it's not specific infection, but person will have constant fever, and the condition is called gram-negative bacteremia, gram-negative microorganisms in the blood. Main symptoms: people feel lousy, they have fever, everything hurts, like you feel when you have flu or something. That makes sense? So that is, and what is more important, you can even kill all microbes. But if there is, you can take LPS, you can isolate LPS, pure, inject it into a mouse, and mouse will have a fever, inflammation, all kinds of bad things. You can isolate LPS and stick it in a human, and human will have all that kind of stuff. Does that make sense to you? This is why we pay so much attention. So this is a typical LPS molecule. I'm not going to ask you about individual parts, so it's more of a FYI. You have a carbohydrate, okay, and lipids. Now, as I mentioned, that's what lipopolysaccharide causes. Non-specific symptoms. For instance, example, one student in the fall, in the middle of a semester, said, "Boy, you were sick a lot when you were a kid." I use examples from, my childhood when I got sick with something, because it's easy to tell you what I felt. I had salmonella once, the mild case. I had terrible fever. I had 39 Celsius fever. Reason for it? LPS got in the bloodstream. Infection was confined to the guts. Okay, it was in my intestines. But LPS from salmonella, which is gram-negative, ended up in my bloodstream, and there we go. I have fever. Okay? Does that make sense? You got LPS idea? Now, before we move on, quick recap. Gram-positives, membrane and thick peptidoglycan cell wall, 2 layers. And in the peptidoglycan cell wall, only gram-positives have teichoic acid, which contributes to infectivity. Here, for gram-negatives, 3 layers, plasma membrane, thin cell wall, outer membrane. In the outer membrane, there are unique molecules, unique for gram-negatives, called endotoxin or lipopolysaccharide, that contribute to general inflammation, fever during gram-negative infection. We're clear. Some microorganisms have slightly different cell wall structures. We're going to wrap up the cell wall story and take a break. For instance, a group called Mycolata, in the study guide, I use different name for them mycobacteria. Mycobacteria is a genus. Mycolata is the order, the bigger group. Mycolata have slightly different structure of the cell wall. Look at the picture here in the left bottom corner. So, plasma membrane and the thick cell wall. What does it remind you of? Gram-positive or gram-negative? Gram-positive. That's gram-positive, right? But outside of that peptidoglycan layer, mycolata have a layer of so-called mycolic acid. Mycolic acid is a fat. It's not so much a fat, it's a wax. Does that make sense? So the cells are basically covered with an extra layer of wax, which makes them really, really well protected against, your immune system, basically. Members of mycolata, the most notable, Mycobacteria tuberculosis. Okay? If you would grow Mycobacterium tuberculosis on the agar, colonies, they look waxy, like they covered with wax. Got it? Good. Actually, some microorganisms lack the cell wall in its entirety. We call those molecules. Microorganisms like mycoplasma. I don't know. Have you heard the name mycoplasma? Okay. Have you heard walking pneumonia? That's mycoplasma pneumonia. Okay. So mycoplasma do not have cell wall, which means the shape is kind of changes all the time. So I tell you, you absolutely must to know the difference between gram-positive and gram-negative in the terms that we just discussed. You must know that if I ask you, okay, what's the difference between mycolata or mycobacteria versus gram-positives? They basically gram-positives, but have a layer of mycolic acid outside. Does that make sense? And for mycoplasma, you have to know mycoplasma does not have cell wall. For archaea, that's FYI story. Archaea do not have peptidoglycan. They have similar stuff. Different glycoproteins, often polysaccharides, but not peptidoglycan. So that is one of the many differences between archaea and bacteria. We got it? Any questions? Anything at all? Okay. We're going to take a break, and when we'll come back, I will print the handouts. Don't worry about them, and we will discuss your results. You can claim your plates right there.